Well, thank you. It's my pleasure to be here on behalf of Argos Therapeutics, a Durham-based biotech company. And as Will mentioned, we're developing the first, hopefully, launching the first precision immunotherapy for oncology, for cancer. So I wanted to start out tonight with this slide, and the theme of this talk is we're all different, and everybody's different. In medicine, however, I don't think we've made enough progress in treating everybody differently. This would be nice. There we go. This is a quote from a local professor, Dr. Ralph Snyderman from Duke. He actually just joined the Argos board. And the basic premise here is one size doesn't fit all. And so there's a lot of technology and a lot of data that has been emerging in the ability to amass big data and also genetic sequencing to really think about personalized medicine as a wave of the future. The White House has a huge initiative on precision medicine, and that's their term for it, and we're going to be using that term tonight, precision medicine. So what is imprecise about current medicine? Well, for those of you who aren't in drug development, this is sort of how it works. On the left side is a trial. So you have a placebo or an active drug, right? And then you test it on patients, and you sort of bucket people into response or non-response, positive outcomes or negative outcomes. And if, on average, your treatment does better than placebo, then you can get the drug approved. But that's on average, right? So you can see the gray patients there. They're more in the positive outcome. The white patients there, a little bit more in the negative outcome. But it's not completely homogeneous. There's some people in gray who have negative outcomes and some people in white who have positive. So then you kind of amplify that potential error, and when the drug's launched and FDA approved, prescribers prescribe it, and 75% of the patients don't respond, okay? And so what precision medicine is trying to ask the question, why? Why do those patients respond and why do those patients not respond? So as Will mentioned, one of the interests I've had, this is a paper that I co-authored last year with some folks from Duke on biomarkers in a different therapeutic area, but biomarkers. What's a biomarker? So it's plasma or tissue. It's something from your body that goes further to identify what's unique about your disease. So for instance, you can have kidney cancer, but this person's kidney cancer is very different from that person's kidney cancer. And that's the premise here is what type of biomarkers exist such that you can identify what's unique about that disease. That's one step. The next step is who's going to interpret that? So you have to have an interpretation, and you say, well, that's different. Let's say it's a mutation. You do a genetic test. What's the norm? What's the database that establishes, is it different? Is it relevant? Is it a driver mutation or is it a passenger mutation? That interpretation is key. The second issue is, even if you do have a biomarker that can phenotype you into, let's say here, just for the sake of argument, yellow, green, and purple, your unique disease, the only thing that matters is whether that biomarker can lead to a precise treatment. And we've made some advances in that. Here you see a genetic test for the patient with a mutation that's yellow, and they have a targeted treatment that matches that biomarker. But we're very far away from having biomarkers for all of these treatments. And matched treatments that match the biomarkers is a huge obstacle that still exists. How many people have heard about immuno-oncology? Yeah. That's basically having the patient, your body fight your cancer. So people have cancer every day, but the immune system fights it off. And what immuno-oncology is harnessing the body's own natural immune system to fight off that disease. It's very different than traditional medicine like chemotherapy or radiation therapy, where it's sort of imprecise, or even drug-targeted therapy, which uses pills, but it's kind of indiscriminate. You have collateral damage. The immuno-oncology field is taking the immune system that exists currently and revving it up, harnessing your own power to fight your own disease. The problems with immune therapy is if it's blanketed and it's not precise, you will have off-target damage or collateral damage. And so some of these therapies, recently, there are a lot of publications out of challenges with toxicities. And I think the third speaker tonight, we're going to talk a little bit more about that. So at Argos, we say, all right, well, if every single tumor is different, every patient's different, their disease is different, what is the key to tumor specificity? There are these things called neoantigens. So these are things that are unique about that patient's tumor. And that's because all mutations are random, right? So everybody's going to have a different tumor evolving at a different mutational rate with different mutational patterns. And so we said, how can we harness neoantigens? Perfect. That was well timed. This is what we do at Argos. So we basically take all the tenets of precision medicine and immuno-oncology and really go after precise immunotherapy. What do we mean by that? Well, neoimmunity without autoimmunity is generating a novel immune response, something that is new and unique to that patient's tumor without the collateral damage of it turning on normal tissue, right? So a very specific response. The two components of our technology are the dendritic cell. The dendritic cell is like the mastermind. It goes around the body and it looks for foreign antigens, whether it be a virus or a bacterium or a cancer cell. And it finds it and then it presents it to the immune system. It processes it. These antigens go to the immune system and says, go look for that. That is non-self. That's a foreign invader. The other part of our technology is mRNA or messenger RNA. It's like the thing that transmits the unique fingerprint of that tumor. So what we do is we try to combine those two things. So how do we do it? We take cells from the patient, so this is called autologous cell therapy. But unlike any other company to date, we actually take the individual tumor from the patient and we take the RNA from that tumor and we load the dendritic cell and we supercharge that dendritic cell and then we give it back to the patient. And it goes back to the patient to restore their immune function and to instruct and arm their immune system to fight that specific tumor. So sort of in sum here, the challenge here is how do you do that? How do you do it one patient, one tumor, going back to that individual patient? Think about how drugs are delivered right now. You go to a pharmacy and there's a common pill bottle that gets your name slapped on the bottle and that's your medicine. But it's really the same medicine that other people are getting. It's kind of like saying, I don't know if you guys saw this ad campaign from Coke. It's a good campaign, but just because your name is on the bottle doesn't mean what's in it is any different, right? So what we at Argos do every day, we touch the patients. We recognize that there's a patient's cells behind that's got to go back only to that one patient who provided that material. I don't know if you can see it here, you probably can't, but it says subject ID number, and what we want to do at Argos is turn that to the patients behind it, right? We know every patient is different. We want to have Harry's Precision Immunotherapy, Ann's, Tom's. It's going back to that single patient. If we're successful, we're going to unleash a firestorm on that patient's cancer. Thank you.